Yes, I have my background in economics, but I have spent the better part of my career interfacing directly with plant scientists, practicing scientists on the one hand, and with lawyers on the other. And that makes for a very interesting combination in an interdisciplinary space that not many people occupy. And what I want to do is use this opportunity to speak to you. I was told by Leah and others here on staff at IGI that many of you are coming from the laboratory. And I want to try to communicate, to teach, in fact, some basic principles of intellectual property and relate them to the specific case of CRISPR-Cas gene editing for crop agriculture, for innovation in agriculture. The purpose for that focus is that this utilization of CRISPR as a suite of research tools, or if you want to call it a platform technology, is going to be governed as it moves from the laboratory into the real world by three sets of factors: intellectual property, the regulatory requirements placed by governments, and foundationally, in fact, the acceptance by the public, by consumers, the social license, as it has been dubbed. Of those three, the intellectual property has proliferated, particularly since the 2012 breakthroughs, of which this campus has become very much embroiled. But we also need to be aware that CRISPR, as a molecular biological tool, as it moves into the world, is confronting the legacy of agrobacteria-mediated transformation of plants, aka GMOs. And therefore, we've got some questions in front of us. Well, the first one that I want to attack is an understanding of the IP landscape. And by that, I don't mean just who's been granted what patents, but then how have they reshuffled and redealt the deck through licensing arrangements. And how does that now govern access to CRISPR for use in agriculture? Then touch on the bigger question of how might that IP and licensing regime end up influencing public perceptions, the broader social questions that we are interested in. And then I'll tantalize, but not really get into a third question, and that is, to what extent might the licensing regime that has emerged in crop agriculture be a model that would be replicable for CRISPR in other fields of use or in other research tools altogether? In order to do this, I want to start out, and I'm going to rip through it pretty fast, but I'm going to give you a primer on intellectual property. And I'll anticipate that doing your biology 101 and all the rest, you've heard of a patent. And so you think you might know a little bit about intellectual property, but I just want to give you a structure for thinking about it, a lexicon so we can communicate, and it'll be useful then in the case of CRISPR-Cas, looking at the patent landscape and the licensing arrangement. And then hopefully we have enough time to really talk about these implications. So let's go. Fundamentals of intellectual property. And I'll start here. For talking about an intellectual property regime, the set of factors that will cause entities to be able to exert control over a technology comes in two parts or two stages. First, there's the granting of property rights. This is the original title as written. But then there's the reshuffling and redealing of that deck, the assignments or transactions or contractual arrangements, what the lawyers like to call the private ordering of the intellectual property regime. Property rights, we don't disagree with property rights. Property rights are okay by most people. Property rights really consist of three foundational pieces: the authority to control how something is used. Second, the right to receive the economic benefits from it, the rents. That's what we call it in economics. And then third, the right to dispose of it, to transfer it, to rent it, to sell it. And in fact, in life, we have basically four kinds of property: personal property, stuff you can pick up and move; Real property, stuff you can't pick up and move. You've got to go to it. Financial property forms, and then intellectual property. So intellectual property is right up there with those others. And there are, in fact, an entire plethora of different types of intellectual properties. And in fact, most of these will come to bear in agriculture and the plant sciences, in fact. Of course, you're familiar with patents. You might not have known that there were more than one kind of patent, including plant patents here in the United States. Maybe you knew that. Utility models are patents light and utilized in many countries around the world, but not the US. But we also have trademarks. You know that it's the US Patent and Trademark Office, correct? So trademarks are administered very similarly to patents and oftentimes cover the same product. We have plant breeders' rights. In the United States, we call it the Plant Variety Protection Office. It's based out of the US Department of Agriculture, not the Department of Commerce. But we've also got data protections, whether it's genomic data or field data, phenotyping data, all these types of data that might matter in your work can be and are protected. Copyright. One that many times we don't think about is the physical property rights. In fact, remember up here, we have personal property. Personal property can bleed over into intellectual property through the ownership of the embodiment of the technology. And in the situation in biology, that's very often the ownership of the physical plasmid or the cell line or whatever it is you're working on. My main point here is for you to understand there's more to this game than patents, but we are going to focus on patents. So patents have, let's think of it as a bunch of teeth. There are a number of different ways that patents can be influenced to be stronger or weaker and give you greater control or maybe looser control. First are the fundamental patentability criteria. There's the subject matter. Is that a patentable subject matter or not? The US Supreme Court kind of changed that for genes in the Supreme Court case in 2015, the Myriad decision, it was called, in which they said, you know what, isolated DNA molecules are no longer, that have a naturally occurring sequence, are no longer patentable subject matter under a particular exclusion or restriction. But you also have to show that your idea is novel, i.e. it's not published anywhere else. You have to show that it's not obvious. That's lawyers' words for saying that it was a bright idea, it wasn't just duh. Yeah, you understand that, right? And then you have to be able to show the utility, that it has a commercial application, that it's not merely an idea. But there are other parts to this that oftentimes are the hang-up for a patentable idea. You need to write and disclose clearly in plain English what it is it does. And you also have to demonstrate that you had it working, at least at the lab bench, your enablement criterion. There are other limitations that can be imposed through patent law. And finally, simple things like patent office rules and regulations, the fees for filing for a patent. I heard up front before the talk began about someone who had an invention but decided not to file because it just was deemed too costly for their project. And that can thwart people from actually taking out the right in the first place. But even after all of these criteria have been met and you're strong on all of them, there's still the aftermarket, as it were, the private ordering. Private ordering can happen through assignment of the patent, and that's usually at time of issue. You write in the name, usually of the Regents of the University of California, for example, if you're employed by the University of California. The sale of a patent affects complete transfer of ownership. So all uses, the right to buy and sell it, the right to take rent for it, go to the new owner. But then there's an enormous, vast space of contractual arrangements that you can strike. Typically, we call this the licensing of a patent. This can be done in part or in whole. You can license just one particular field, like, let's say, the use of CRISPR in agriculture, or even more narrowly, the use of CRISPR in small grains. You could do that. You could write a contract that would say that. You could also designate just research use. or commercial profit-oriented R&D, and split those out, and that is typically done. Your contracts can be time limited or they can be in perpetuity. You could put secrecy and non-disclosure requirements around an agreement. You could have all sorts of funny conditions on, if you make an invention using my invention, then you need to give me rights to use your invention too. That's called a grant back. Maybe I get some royalties off of your invention. That's called a reach through. You can put all sorts of other constraints and conditions, just about as much as you might imagine. As a molecular biologist, think of this as the building blocks, you know, the codons, the genetic code or whatever, of a vastly complexifiable creative space that lawyers will take and run with if you give them much of an opportunity. Now let's take this basic lexicon around intellectual property and start looking at research tools. Inventions with a specific set of characteristics, and by the way, CRISPR and genome editing is one of these. And the first important thing that we need to see is that there are pretty high fixed costs in discovering or creating this tool, and a lot of uncertainty. You don't necessarily know that's what you're going to come out at the end of the research project with. Yet once this invention is in your hands, it reduces the costs of making follow-on innovations, which themselves might be stochastic or uncertain, and it expands thus the feasible set of what you can work on now. Now, if you don't remember anything else I say today, I want you to remember this line. This is one of the most important aspects of a research tool such as CRISPR, and that it has a dual nature. It may be used purely as an upstream tool, enabling further research, but it may also end up moving downstream through the development process and become embedded in a final product or process that goes onto the market. And it's because of that duality, it's because of that split that we very often run into policy troubles with research tools and the administration of intellectual property around them. Clever private ordering or licensing can help to manage this. And I hope to be able to demonstrate to you that in CRISPR, we've done a reasonable job so far. But more about research tools. There are also many potential users of the tool. Therefore, to maximize social welfare, let's say, to make the world as best a place as possible, or to come up with as many useful innovations as possible, you want to make it broadly available so that everyone who might have a bright idea, they might have an idiosyncratic capability. They can do something you can't do if you're the one who owns the research tool. They might have privileged information, or they might just have a bright idea. So this could and should be treated as a public good, but in economics, we call this a quasi-public good, when it's not just purely something that we hand out to the entire world at zero cost. Now, these many users may be competing in the same market, or they may be operating in completely different niches, different markets. And it's hard to anticipate up front what that demand is going to look like. And finally, typically, research tools can be quite complex. In other words, there's not just one atom bomb of a patent that covers the entire technique, that rather it's broken up. It's a fractured property rights regime that operates to protect. And this often is dubbed a patent thicket. If there's 5 or 10 or 20 or 40 or 100 different patents that may read over different aspects of the use of a suite of tools, now you have a coordination problem on your hands, potentially. It's also been called the anti-commons. Let's look at a few key biological research tools here. Can you read that? First, I'm throwing up the transformation technologies. 1973, Cohen and Boyer out of Stanford and UCSF. Stanford and UCSF took patents on the Cohen and Boyer invention, And they broadly licensed it non-exclusively to well over 400 different companies. They received quite a bit in royalties back for that. The axle patents out of Colombia, but also agrobacterium. It was a three-way race for the patenting of agrobacterium between Monsanto, Washington University in St. Louis, and University of Ghent. Other research tools that have been patented. In gene expression, there's antisense and RNAi. Again, some interesting patenting situations around those. Stem cells represent another area of research tools where we've had some interesting back and forth. And then we come into the world of gene editing. And of course, CRISPR-Cas9 is just one of multiple different methods of nucleases being used to target and edit DNA. A couple of initiatives that David Zilberman and others here at University of California and myself have been engaged in involved a couple of these research tools. One was called PIPRA, based out of UC Davis. PIPRA still exists and is helping scientists to understand how to patent and license their technologies. But in its early days, we were seeking to help coordinate university-owned components of agrobacterium transformation and make that available for research and for commercial uses. And an initiative around the access to not just the intellectual property, but the biological materials, as well as data on the provenance of stem cell lines, is something that several of us participated in the mid-2000s. So now let's move on to the CRISPR-Cas platform. You have a sense, I hope, of what intellectual property is, how it works, especially around biological research tools. At least we have some language. So now let's delve into the CRISPR-Cas platform. And the first slide I'm going to bring up here is kind of a heinous graphic. This grew over several years as the granting of the rights unfolded. And I've run out of space on the right-hand margin, but there's more to the story. But suffice it to say that around 2012, we had essentially 3 major patent applications going in around the original breakthroughs. Those from here at UC Berkeley, with some familiar names probably to those of you who are here. MIT with Feng Zhang, and then Virginia Sixness at Vilnius. All of those original applications went in 2012. Yes, 2012 for all of them. The patents were not all issued until ultimately the Berkeley patent came out in June of 2018. But what essentially you should take out of this is what emerged from this were 2, and in fact, potentially 3 university patent estates over complementary different core components of the CRISPR-Cas9 system. There's no one atomic bomb patent that is all of CRISPR, and we all acknowledge that. How the details of these conflicts work out is still being written. Obviously, the conflicts have played out differently in the United States and in the European courts, and we will see. But from the point of view of a user, it, in the end of the day, doesn't really matter, because what you are looking at as a user is the broader accessibility to the technology. Now, if you're in the university, you're probably not too worried about it, because the plasmids and the reagents were quickly made available for you to use. If you go to Add Gene, you put in your order, you can get a shipment. It costs like $60 for them to FedEx you a particular plasmid. Now with that, you are executing an agreement. This is part of that private ordering that I told you about. You are executing an agreement for them to transfer those biological materials to you, and you were agreeing to a set of conditions. And the important part is it's for academic and research purposes only. So you can do your dissertation, no problem. But if you decide to patent and start a company based on what you did your dissertation on, this isn't going to cover you anymore. Your rights have ended at the door of your laboratory. And as soon as you start your startup, you're going to have to go back to the source and talk to them about commercial uses. But this has been a roaring success. Adgene has shipped, nowadays it's probably over 50,000 plasmids to entities all over the world, and it's their single most popular material that they run out of this plasmid repository based in Cambridge. Now, there's a wide range of uses of CRISPR, obviously, across the biological research. This is a plotting that I just pulled out of Web of Science, showing you the number of papers being published through, it's current through the end of 2017. But this is illustrative of the success of the Adgene exchange of materials, enabling basic research using CRISPR. Now, interestingly, not just the three key portfolios of the initial breakthroughs, but follow-on patent filings have followed a curve that looks like this. And this is some unpublished data that several of us went and ginned up a couple of years ago. At the end of 2017, we had close to 2,000 inventions worldwide using CRISPR on some component of a CRISPR system, some additional tool, a new guide RNA, you name it, different pieces of it. And these are filed all around the world. This image here shows you that the most prolific and earliest set of filings were in the United States. That's the high set of light blue bars at the back of the graphic. The red bars, the next one up, is China. And it almost looks like a two-horse race when it comes to where the filings are happening around the world in CRISPR. After China, we've got the European Patent Office, United Kingdom. These are the top 10. And as you can see, much, much less activity going on at these national patent offices, but even at the European-wide European Patent Office. If you are using CRISPR in, let's say, the Philippines, working on rice, you are probably not subject to CRISPR patents within that jurisdiction. And remember, a patent is a national or a regional right. The constraints of the patent end at the border. And so if you do the work outside of the jurisdiction of the United States or China, you are probably not even infringing. If you ship a product into the United States or China that used and embodies a CRISPR component that is patented there, that's when you might start getting into some trouble. With this data, we can also analyze the assignment. Remember, patents are assigned as part of the private ordering process. And those three patent estates that I demonstrated the breakthroughs to have occurred in have come together in the full data set. The MIT, Harvard, Broad Institute guys have the most by head count. These are actual inventions, and you can see that there are several hundred coming out of Cambridge. University of California tallied 82 CRISPR inventions by the time we had compiled this data. Editas and General Hospital, that's part of the Cambridge crowd. And then we have Pioneer Hybrid. Yes, the seed company. Pioneer is owned by DuPont. DuPont and Pioneer have also licensed in all fields of use exclusively the Vilnius patent. Let's start digging into some of these aftermarket ordering issues. That's how things came at assignment. Now, how have the core inventions been reordered and, more importantly, made available for use in crop agriculture? So now's where we kind of descend into the soap opera part of the talk. And I want to take about 3 slides here to walk through how each of these 3 states has shaken out a little bit. And you guys can probably help me on my first slide here because I probably still don't have the University of California story all figured out. The lead inventors that show up in the press all the time are Doudna and Charpentier. In 2012, that initial single patent application went into the US Patent Office with 155 claims written in the patent. The claims are the individual statements of what it is we did and what we claim to have rights to. I don't think I can pull a good average, but it would be somewhere on the order of 25 to 50 per patent is what it gets typically written. So this was a very large, very broad patent, piled an awful lot of claims into one document. And that actually may have become an issue in a while. Now this patent was a little bit interesting given the co-inventors here at Berkeley. The assignment of rights went first of all to the University of California under the Bayh-Dole Act set of rules that we operate under federal funding in the United States under. It also went to the University of Vienna because Austria has a similar set of rules. And Emmanuel Charpentier had a split appointment at the time between University of Vienna and the University of Umeå. Can you say Umeå? In northern Sweden. Sweden, interestingly, has a policy that grants the rights to an invention directly to the individual faculty member. And so Charpentier was able to take an assignment as an individual, which put a very interesting twist on the story here in California. One thing that she did was she founded a couple of different companies, but ERS Genomics was given an exclusive license to her shares in all fields other than human therapeutics. Remember, you can license in different fields, and that's the way it was framed. This will include all agricultural applications. So ERS Genomics becomes the holder of Charpentier's rights. Oh, and I forgot that little bit. Of course, you're probably familiar with Caribou Biosciences, which is a nearby biotech company here, which Doudna founded. And at least to the initial inventions, UC Berkeley gave exclusive license to Caribou in all fields. So now you have a couple of surrogates, startup companies that have inherited the rights to the UC invention. Next, we're going to turn to DuPont. DuPont is watching this space. They've owned Pioneer. They've operated in ag chemicals for a long time. And they have spent 20 years or so getting their hand burned by one of their direct competitors in the space of biotechnology and agriculture. I won't name names. So they moved aggressively early in order to assure themselves freedom to operate. And the first step they took was invisible to most people at the time. But in 2012, at the same time that things were just breaking here in Berkeley and in Cambridge, DuPont bought Danisco, which is a little yogurt dairy company in Copenhagen or Aarhus or somewhere in Denmark. And there were several inventors there that had done some earlier breakthrough work in CRISPR. And not only did this give DuPont the rights to those patents, but it also gave them an inside track on information and some in-house expertise in the technology. And those individuals in Denmark introduced them to this fellow way off the edge of the world, right, in Vilnius, Lithuania, who was also a leading member in the science of microbial defense systems using CRISPR, Suxness. DuPont showed up early and in 2013 made a very strong offer to the University of Vilnius for the rights to that patent. DuPont then came to Berkeley and knocked on the doors at Caribou, negotiated for a couple of years, and ultimately signed a cross license with Caribou for the University of California patent rights in the areas that DuPont cared about, and of course offered Caribou some intellectual property in return. Whoops, things are a little out of order, so let me just bring it all up. Well, you can read the details, but let's jump on to the final piece in the DuPont circuit here, and that was in 2017. They then approached DRS Genomics in Europe for rights of Charpentier's piece of the University of California invention for uses in agriculture. Oh, and one last little bit, and I'll highlight this for you again. In 2017, really the announcement was first made in 2015. Dow-DuPont was announced, the merger of Dow and DuPont. And Dow had some activity in this space as well. So Dow has worked to aggregate up a portfolio here, as you can see. And then the third estate was that of the Cambridge gang. The Broad Institute became the lead manager of the inventions from all of the co-inventors there. The Broad Institute is technically a joint venture between MIT and Harvard, and some of the teaching hospitals, and all of the faculty at the Broad Institute also have a faculty tenure track appointment at one of the home institutions. The initial filings, they basically took a very different strategy with the initial invention. Instead of filing one patent with 155 claims, they filed 11 patents, probably with an average of 15 claims each. So instead of publishing it as a book, they scattered it out as a whole bunch of journal articles. Would be one way to analogize the situation. Now, these were all assigned to the Broad Institute or MIT or Harvard or the collaborators' home institutions. And some additional inventions followed. And these are all details, but they include a number of the New York City and other East Coast universities. Now, the Broad moves very quickly in licensing out. I sat down and talked with the head of technology transfer for the Broad Institute about a year ago about their approach, and he stressed that they are really trying to license under what they call an inclusive innovation model. Now, this inclusive innovation model does allow them to license exclusively, but it limits it to a very specific target, a very specific field, and it's time delimited to just 2 years. And if you cannot show that you now have a product ready to go, and this is very short, especially in therapeutics, they will go ahead and sunset that license on you and bring back the rights and assign it somewhere else. And they were doing field-specific licensing of CRISPR quite early on. Of course, I already told you about making it available via AdGene for research use. They then made specific commercial licenses, non-exclusively to Sigma Aldrich, GE Health, Horizon, the other big reagents companies, so that you could order it through the catalog. In human therapeutics, they made a field of use specific exclusive license to the startup company Editas. So now you have another surrogate biotech company that has inherited a very broad piece of the core. And in all other fields of commercial use, including agriculture, they started granting dozens of non-exclusive or limited exclusive licenses under this so-called inclusive innovation model. So they were already, in fact, one of those was Monsanto. So by 2017, the world had evolved to look something like this. You had the 3 academic estates that DuPont Pioneer had started taking steps to begin to aggregate and bring together through their field-specific cross-license with Caribou, their exclusive license, at least for agricultural uses, from ERS Genomics, which came out of the Charpentier share of the Berkeley patent, and they had the exclusive to the Vilnius invention. They had merged with Dow, and they were beginning to license out. And then over here we had the Broad Institute with the startup Editas doing human therapeutics, but these range of field-specific non-exclusive licenses for agriculture, including Monsanto. So which horse are you going to bet on here? The Monsanto horse or the DuPont Pioneer horse, the Broad horse, or the broader mess that we basically have over here from the private ordering that has gone on so far? This was posing fodder for all sorts of commentary about the difficulty of potentially moving any sort of major commercial product to market in agriculture. And then in October 2017, this press release came out that DuPont had been talking with the Broad Institute and that they were joining forces on a joint licensing framework, as they called it, to provide non-exclusive licenses to CRISPR-Cas9 for commercial agricultural research. Do you read that? Commercial agricultural research. So this, we've already got Adgene making the plasmids and the reagents available to us in the academic laboratories, but now there's this offering of essentially a pooled license that brings together the entire set of patent estates. Now, just to remind you about this little change, the Dow-DuPont merger was first announced in 2015. They then rebranded the Pioneer and the Dow AgroSciences merged divisions as Corteva AgroScience. It was a division of Dow-DuPont from 2018 to 2019. And in June of this year, Corteva went basically through an IPO and spun off from Dow-DuPont to become a standalone company of its own. And it has inherited through that divestiture, that spin-off, the CRISPR rights for uses in agriculture. So sitting down with basically the two managers that developed this joint licensing framework about a year ago, they laid out for me the essence of what they've done here. The motivation of the parties, as they report it anyway, we can speculate that there might have been other strategic and even political reasons behind it. But they say they wanted to help accelerate innovation in plant agriculture, and in particular to promote social license with broader adoption of the technology. And that this would provide efficient access to what so far had been split patent estates, roughly now the Broad Corteva patent estates. Now they acknowledged every time they sat down to cut a license with one of their own licensees to their own estate, they said, now we can guarantee you that we won't sue you, but we can't vouch for the other guys. So you might want to go talk to them too. But that introduces a whole set of strategic challenges to the industry. Yes, they said that they could have let the courts decide which patent is the valid one, but most managers of R&D in industry don't want to either A, sit around and wait for the courts to hash this out. That could take years. And B, they would rather hedge their bets. So they were attempting to go to both parties and negotiate already. So this was a way, in essence, to streamline that process. The licensing framework, now let's just be very clear about this, is that it's field specific for use in crop agriculture, not in animal agriculture, not in dairy, but in all fields of crop agriculture. Both parties put in the relevant IP rights that they had at the time that they controlled at the time for practicing CRISPR-Cas9 in crop agriculture. It covers about 40 inventions. They told me the number, but they said they have not published the list, that is privy to the licensees. It includes Cas9. It focuses on the use of Cas9, and that's intentional, they said, not only because that's what they had the most control over, but also they envision synergistic benefits. If everyone uses Cas9, that it'll lead to further improvements in the technology and its efficiency, but that it will also, if you will, cut trail through the regulatory arena. that the first few products that go through will help to set precedent, and then that will make it easier and cheaper for follow-on products to get approved. Now and we must emphasize there's no agreement or mechanism to refresh or update the technologies in the pool. In other words, everything that has been invented since 2017 isn't necessarily added to it. Now, the first initial reflex by those of us that watch this arena was to almost call this a patent pool, but it's not. It's similar in its results and the outcomes, but technically it's a joint license. And in fact, what it means is you're signing two documents, one with the Broad Institute and one with Corteva, but the terms are all written in such a way that they complement each other. Not too terribly unusual in the legal world to have complex licenses of that nature. And these two parties are representing all of the other original assignees of all 40 patents that are in their control and that they put into this. Now, there are some limitations, some of those controls and restrictions that I mentioned you can have in a licensing world. One is no gene drives. And that's partly just because of the publicity that I think had gotten ginned up quite early on in the CRISPR conversation about gene drives and how it enabled those, and many people in the public started to conflate the two, and these guys just wanted to set gene drives aside, even though there could be potentially valuable applications in agriculture. No tobacco for consumptive use, so we're not talking about tobacco as a model organism here, and also no seed sterility or terminator technologies. These are the three big limitations. Coming from Colorado, I had to ask them the obvious question about cannabis sativa, and they pointed me to their next two obligations. One is that you have to have a stewardship plan in terms of getting it through things like federal regulators, and secondly, compliance with all applicable laws. That's a really nice general clause that a very smart lawyer came up with in order to dodge the cannabis question. And they said they are actively licensing to direct competitors in crop agriculture. We, in fact, just had a meeting in Keystone, Colorado a month ago. A couple of folks in the room here were involved in that. And we had Bayer Monsanto in the conversation right there along with the Corteva folks, and they're acting like old friends. The licensing framework had the initial idea of being a simple click-through clearinghouse, operating somewhat like the add gene mechanism where you could just hop online and say, let's do it. But what they found was that, in fact, the users were wanting to customize and not pay for stuff that they weren't going to use. I'm not going to go into the details here, but there are a number of different flavors of the kind of license that you can get. And depending on the flavor that you get, you will pay for it in a different way. And in fact, they have all of the major possible royalty mechanisms, including an upfront fee, a fixed annual fee, milestone payments that depend on how you get there, and a running royalty as a percentage of the benefit created by the technology. And the most important thing that they emphasize as they lay this out is that all of these are on a sliding scale and depend on the size of your company and the market that you're going into. This allows for flexibility in the negotiations. Some of these can get zeroed out, and they said the intention is to make sure that the costs are not prohibitive. They're trying to be fair, and they explicitly use that word. And they also pointed out that you would not be at all subject to royalties if you were trying to develop something for use in a developing country benefiting smallholder farmers. They've been approached by over 70 parties, and the first licenses started rolling out late last year. So let's go ahead and wrap up. Just to conclude and summarize, academic uses of CRISPR for agriculture are already having an impact. The Add Gene Access has enabled a tremendous amount of research that is being published, but that will also inevitably be leading to developments that will be useful in commercial agriculture. Those uses, as we started, are going to be governed by the IP together with the regulatory and public perceptions. And until 2017, it wasn't clear at all how you were going to get through the IP portion of that maze. But with this new, essentially public-private partnership, the joint license from Broad and Corteva, there is now significant potential and access basically for anyone willing to sit down and negotiate with them. Early indications that it is being licensed broadly. So now let's end right back where we began with the big questions. Is this going to be enough to, in effect, democratize innovation in agriculture? In other words, to bring a suite of products and innovations to market from companies, big and small competitors, different verticals within the industry, enough so that the public becomes familiar with it and comfortable with the technology. And will that turn public opinion? Will that end up politically weighing into the regulatory arena enough so that it makes the use of the technology ubiquitous? And if it succeeds in doing that here, to what extent is this a possible model for collaborative licensing regimes in other uses of CRISPR in other fields of use? And with that, I'm going to go ahead and tie it off and say thank you, and certainly take questions if anyone has any.
So in our new tradition, we'll take the first question from graduate students or other students.
Ah, good tradition.
I'll wait.
Sure. So you talked about how this could potentially influence the regulatory, the way that these get through the regulatory process. Are they trying to, in this broad licensing agreement, part of their selling point to individuals who license with them will be like, we will blaze this path and allow the, we'll work on the regulatory stuff, you can work on the actual innovation?
I don't believe it's explicit in the license language, but it's implicit in the logic, if that makes sense. In other words, hey guys, let's all take this path. And if we all walk this path together, we'll beat more of a path. Whereas if you go and use that enzyme and you go and use that enzyme, regulators have to go and evaluate the safety of each of those independently, spreads out the resources of the regulators, whether it's FDA, EPA, USDA, whether it's the European regulators. You're going to have to probably get it through all major trading partners, by the way. That's the way agriculture as a commodity business works. So the Canadians, the Australians, the Chinese, wherever you export or wherever you import from. So it's actually a very big economic argument for trying to streamline as much as possible there. Now, granted, the major regulators like the US and Europe tend to have influence, if you will, over regulators in other countries. So once the US has approved a particular product, I mean, there's always tit for tat or, oh, we need to assert our own sovereignty and we'll go ahead and reject something that you guys approved. But generally, other regulators will look through the decision that the USDA or the FDA take, and that will inform their assessments as well. That's the logic of this, is an implicit teaming up and traveling the same path together, if that makes sense.
Go back here.
So you mentioned in the final slide that this might be a good framework for the future of other applications for this system. So there are already a number of CRISPR therapeutics in clinical trials. So what is the process for someone looking to turn that into a commercial product at this point? Like, what would that look like conceivably?
A therapeutic in particular.
Yeah, to basically, if someone wanted to get money from using this treatment commercially, what would be the legal framework they could do that with without exposing themselves to lawsuits from, you know, the 3 estates, basically?
Right. Well, you could be ambitious and you might find a willing ear with the patent holders to say, hey, we would like you to set up a joint licensing framework for uses in our therapeutic indication in pancreatic work or whatever it might be. Think about it though. How likely is that, right? What I was intoning was that maybe if you're doing work in tuberculosis or malaria or HIV, in some of these areas where we have had orphan drugs, particularly in the infectious diseases area, I think that's your next logical area where the parties that own these patents might be willing to sit down and put the whole estate together and offer it under one set of negotiations to multiple parties. The other key to this, if you remember back to the economics of research tools, is that to warrant going through all of the wrangling that it takes to set up a joint licensing framework like this, you need to be, in essence, offsetting what would have been a whole bunch of negotiations with separate downstream parties. In other words, you need to have enough demand out there, enough competing or collaborating players, all of whom need freedom to operate with the tools to warrant going through the fixed costs of setting up the licensing framework. So if that's what you mean, then I would say, yes, it's probably most likely in particular applications of that nature. But the bioeconomy is big, guys. And you probably know better than I. You're one of those other potential users out there with privileged information and bright ideas. If you've got an area that you think warrants freedom to operate of this nature, start raising some cane and talking about it, because I would say these guys are open to discussion. Go and talk to Matt Mueller at Corteva Agrisciences, the head of their Open Innovation Division. Go talk to Issy Rosen, the head of business development at the Broad Institute, and say, here's my idea. And they'll at least give you a little bit of an ear and tell you why it won't work, right?
So the most common way to deliver Cas9 into plants is using agrobacterium. My understanding is that the agro patents, even though they're filed for 35 years ago, are still active by Monsanto. Are they part of this joint licensing agreement, or would that be separate? Are there any other patents that are important to actually use this technology?
This pool gives you the core CRISPR and Cas. It does not give you delivery. It doesn't give you any of the other add-on tools. So it is fairly focused. And that is to emphasize that absolutely there are going to be other complementary technologies in the broader suite that you may need to go and figure out separately. The benefit here is just that what could have been a nasty set of negotiations around the core have been simplified for you. As far as agro goes, I'm a little bit off my game in terms of watching expiry on all of that. Some of the agro patents are now back in the public domain, but you're right. You need to tread carefully there in terms of using agro. If you have a different delivery mechanism that's feasible and you're serious about developing something for crop agriculture, either get a license from them for that. Bayer may be open to that conversation at this point, or find another delivery mechanism.
So we'll do one more, and then as usual, we have the lunch out there. So more questions, come sit, and you can ask your questions at lunch. So for the last question, we'll go to David Sullivan.
I think the key assumption is that there is a public cash to biotechnology. The reality is that it's a political situation that there are a small number of groups
Right.
And the same, if you look at the same number, the 20% that oppose GMO, the other 20% that oppose CRISPR.
Yep.
And so to some extent, this mechanism basically is designed to say, gosh, now we don't have a problem with intellectual property.
Right.
So to some extent, how do you see this arrangement try to deal with the fact that we really have a political processor? Because the big issue now is the role of CRISPR in Europe, and basically in Europe, CRISPR was defined as another form of GMO.
Right.
So how does that try to deal with it? Does it speak about it, or they maybe say maybe we'll be able to get a better deal in some in China or in Japan or something like this? How does it deal with the issue of Europe?
The way that I interpret it, let's look at it in two layers, okay, David? There's the populist view, the public perceptions of. If we go back to the world of agrobacterium, it was pretty clear that the research tool itself was being used as, if you will, a weapon of industrial competition between the majors in crop genetics. And the weaponization, if you will, of molecular biology is a big part, I think, of what then spilled over into the public perceptions of GMOs being suspect. It was collateral damage that some of the competitors were willing to take. And they didn't do it with probably a lot of foresight. They were, competing in the ways that they had competed in, pesticides markets for decades before that, right? Now, essentially, you've got these players, or one of the players who has clearly moved, taken a dominant position, calling détente and saying, we're not going to use the tool as a weapon of competition with you guys. In fact, here, you can use it too. And they're directly licensing their competitors on the calculation that that will help mollify the politics at the regulatory agencies. It will keep the little guys, AKA the biotech geniuses who would do a lot of the innovating and stopped innovating, by the way, with agrobacterium, right? We'll start inviting startup companies to work with this again, recognizing that they might be able to get a product into the market. And then the calculation is that if it starts becoming ubiquitous from multiple innovations coming to market, that the Europeans will start to recognize that, wait, there's not a looming threat here. There's benefits, there's maybe some doubts, but it's nothing different than most other technologies in the market any longer. Whether or not those politics are already, that die may already be cast within European politics. You may have green parties that, have a specter out there, and they will continue to use that as a way to garner votes and tell people that we're going to protect you from the threat of, any biotechnology in your food supply. As long as enough people believe in that meme, it will maybe persist as a political mechanism. That's, however, the uncertainty, the calculation that's, I think, going on in this. Does that make sense?
That's why human therapeutics might be better stuff. No one's going to stop.
Well, and the human therapeutics are going to happen. They are happening regardless. Yeah.
So let's continue this conversation at lunch, and let's thank our speaker.
Absolutely. Thank you all very much.